Since I was a little girl, I've been fascinated by biology. There's something magical about teeny tiny cells banding together to give rise to something that transcends their individual beings. Something with really complex behaviors and architecture, even able to explore itself and its individual parts. So, yeah, safe to say no one was surprised when I decided to become a researcher. More specifically, I was drawn to the effects of radiation in living organisms, which led me to Chernobyl. I've worked there since the early 2010s in my team, and I have explored a good deal of the restricted areas. This story, though maybe if you asked Luca he'd say I should use nightmare, began February of last year, when a Russian drone struck the new safe confinement structure and left a big hole on it. It all started because the radiation levels we expected were not matched with the actual readings. It's not like I study nuclear physics, but you can probably guess that, in my line of work, Geiger counters are one of the most basic tools. It was definitely strange seeing the radiation level had diminished that much, almost a whole order of magnitude. It was quite a dramatic drop, but the institutions didn't seem bothered for some reason. I guess the war made it so this wasn't a priority. It was for me, though. So our team sent a small, remote-controlled rover of our own to investigate the insides of the nuclear plant. The vehicle carried a Geiger counter, cameras, spectroscopic gamma, thermal imaging, regular ones, air sampling unit, LiDAR. The whole package. We had never been able to access certain areas of the nuclear plant due to the absurdly high radiation, enough to completely destroy anything, biological or artificial, in a matter of minutes, if not seconds. This time, however, the rover's Geiger counter told a different story. This time radiation levels had dropped to somewhere between 3 to 4 millisieverts per hour, still high, but within the acceptable range where a suited entry of 2 to 3 hours wouldn't exceed annual exposure limits. The whole team was on edge, not only because of the readings, but also because no one had been able to see what lay down there. Me and 2 other researchers standing still, eyes fixed on the screens. The rover kept going through the labyrinthine basement corridors under the reactor itself. Some were flooded, some collapsed, but there was a tight hallway big enough to allow the rover to pass through. We were exploring a place no living person had set foot in since the nuclear disaster. "Look, over there," said Johann, pointing his finger to the screen with the spectroscopic gamma camera readings. "These cesium-137 and strontium-90 signatures are weird." They were indeed strange, as the readings showed they were completely localized instead of dispersed. For decades of dust, air and water flow should have spread the contaminants almost evenly, and yet we were expecting to discover some sort of radiation-eating organism, or that was the initial hypothesis the whole team was playing with. There had to be some reason these isotopes were bunched up in a specific area, and, our best guess being biologists, was that there was something feeding off them. It made sense, right? "It's just behind that door," said Luca, tapping the optical camera screen. There was a rusted door that led to a room adjacent to the bubbler pool, which used to act as a pressure suppression system and emergency coolant reservoir. "It looks frail enough to get in if you push it with the rover." So I did, and the door didn't open, but collapsed inside the room as if it was barely holding while standing still. The first images were confusing, to say the least. The optical cameras and illumination from the rover were obfuscated by a thick layer of floating dust after the collapse of the door. But there was definitely something wrong in that room. The geometry felt strange, with weird-looking blobs stuck to the walls. There was a red tint to it, almost crimson. It was a vivid color that contrasted with the dullness of dust, dirt, rust, and concrete. The texture seemed slimy, humid but not really wet. The LiDAR readings showed the growths anchored to the walls, with concrete apparently dissolved underneath them. "Anastasia, could you change the view to the thermal camera, please?" When I did, the whole room lighted up. It was hot. Walls, floor, and ceiling, everything painted in red. The readings showed temperatures ranging from 35 to 39 degrees Celsius and some sort of liquid constantly flowing all around the room and whatever the blobs were. "That looks like a circulatory system," I said. "And the temperature, it's quite characteristic, almost like it's alive." Luca and Johann looked at me, not frightened but almost excited. It was definitely biological in nature, and the spectroscopic gamma readings confirmed the cesium and strontium isotopes were densest inside that tissue. They were even more concentrated inside the most distended structures. I'll be honest, there was something unsettling about this whole thing, but so far it was everything we were hoping for, wasn't it? It certainly looked like a living organism that fed off radiation or, at least, radioactive isotopes. We should start taking samples, said Luca, trying to contain his excitement. I'd say we have to try to find out if it's a single organism or some sort of colony. Take samples of different parts, like... Could you get the rover a bit closer to that wall? Johann didn't seem as happy about the finding as Luca. I complied and slowly pushed the joystick forward, commanding the rover to move several meters towards the wall, still using thermal imaging. Then switched to optical. I wanted to take a good look at the blob thing before we took any samples. Is that a foo / intestine? Luca covered his mouth. It's bloated, could it be gas or... I switched to the thermal camera once more and verified the temperature was homogeneous throughout the whole blob, intestine or whatever it was. If it was indeed filled with gas, the temperature should be lower as gas has poor thermal conductivity. Even more, the blob was almost half a degree warmer than the surrounding tissue. Both my colleagues arrived at the same conclusion with no words needed. Then Johann placed his hand on my shoulder and, with trembling voice, asked, Is that moving? There was movement inside the blob. Inside the pseudo-intestine. Not movement like that of the fluids captured by thermal imaging, free-flowing inside the organic network. There was something solid that didn't follow the fluid dynamics of the surrounding circulatory flow, more rhythmic than turbulent. The temperature suggests an elevated metabolic rate, I said, not wanting to continue. I was afraid of what I was going to say next, like that of a pregnant woman's womb. What the f / are you trying to say, Anastasia? I turned and looked Luca right in the eyes. He knew exactly what I was saying. His excitement had turned sour, to something darker. There was a subtle hint of fear camouflaged under a layer of incredulity. Just, take the sample, will you? I drove the rover back through the narrow corridor and out of the immediate radioactive zone, then stopped it at the designated decontamination point. Johann suited up in his protective gear and sprayed the vehicle with a decontaminant solution, then scanned it thoroughly with a Geiger counter. Any reading above background would lead to another full decontamination round, but the low radiation levels worked in our favor this time. Then he removed the canisters with the organics while still in the decontamination zone. Johann manipulated them carefully, sealing them in multiple layers and containers, like a matryoshka doll. The last step was placing them inside a lead-line transport case labeled with radiation warning tape. When he was going for the canisters containing the air samples, he stopped. Are you watching this? He asked, pointing the Geiger counter towards the air sampling unit. It looks like dried blood. Dried blood and filth. It's occluded. The air canisters feel. Johann shook the one in his hand and it emitted a sound like it was full of liquid. They definitely have something liquid inside. Over the next 3 weeks we carried out several different analyses on both the tissue samples and the air canisters. A radiological screening to assess the radiation levels and determine what containment was needed for lab work. The results showed there was an extraordinarily high concentration of cesium-137 and strontium-90 in the tissue samples, which meant they would require handling in specific containment environment. Then, once we knew how to safely work with them, a basic histology study I examined them under a microscope after preparing the sections and staining them. At this point, I didn't know what to expect. I was already unsettled by what we had seen and theorized, but nothing could prepare me for what I saw under the lens. The cells were human. They were unambiguously human, enough for me to instantly recognize them. Their nucleus, cytoplasm, organelles, all human, but with expressed proteins and certain structures that were heavily mutated. And the type of tissue was clear enough to me once I knew their nature. They were cells from the uterine lining. We had taken samples from a womb. A living human womb. The genetic analysis was no better. If anything, it was even more unsettling. PCR and sequencing. The DNA was heavily mutated, with enough genes intact to establish the cell line. The results of mutations were coherent with those found in certain kinds of aggressive cancer. The methylation patterns showed systematic silencing of DNA repair pathways. Genes that would help repair radiation damage had been progressively shut down, stopping the cell from fighting the mutations and just collecting them. It was as if the thing was building itself out of the radiation. The mutations resulted in new genes that encoded new proteins. And some of these were functioning as novel enzymes, catalyzing reactions that normal human cells doesn't. There was some deep coherence there, not just a random tumor. Cancers do not have this level of functionality, rather they just hinder the correct functionality of that which they taint to a pathological level. We had discovered something transcendental. Well, if I wasn't convinced then, I sure am now, said Johann, arms crossed. He wasn't amused, his tone was dead serious. That thing's alive by its own right. It's not a tumor. It's not a person either, started Luca, then his voice broke a little, right? None of the samples contained anything resembling mature neural tissue, I replied. Only uterine. I paused for a second as I remembered something, though several samples showed anomalous concentrations of cells expressing modified versions of mechanoreceptor proteins. It's, the same proteins involved in pressure and stretch sensation in normal uterine tissue. Silence. What about the air samples? Johann raised his tablet and unlocked it. There was an unopened message on the inbox, subject air samples 33 to 37 test results. He had received it barely 5 minutes ago. He downloaded the attached file from the external lab that analyzed the canisters and opened it. The canisters contained particulate matter at concentrations approximately 40 to 50 times higher than background levels in the outer corridors. The room's atmosphere was dense with suspended materials. The particulate analysis identified it as a biofilm, included. It was composed of 3 components: radioactive dust consistent with the known isotope profile of the basement environment, fragmented lipid membrane material consistent with lysate of biological origin, and intact organelle-like structures. More specifically, what appeared to be some sort of modified mitochondria more consistent with active aerosolization than with mechanical dispersal due to their concentration. The report also included a comment pointing out that the modified mitochondria were still metabolically active at time of analysis. 3 weeks since the sample retrieval. I think we should report this, Luca mumbled. I don't care if any other research group takes merit. This is too. Like, what the F // does that mean, Anna? Johann was about to freak out, same as Luca, but at least he was trying really hard to keep it together. Take a breath, Luca. This is exciting. Right? A new discovery, something that nobody but us knows exists. As something to study. New? It's made of F / G human cells, Johann. It's alive. It's been there 4 damn decades. Maybe it's better if nobody knows it exists. There's no word for whatever that is. He pointed to the video of the optical cameras of the rover being replayed on a background screen. It's a human uterus, disembodied, somehow. And it's not just one, the whole room is covered in them. They're eating the concrete, the foundation of the plant. It's almost as if they fed off the radiation, like F / this. Lucas stormed off the room. A day later, the samples and all our equipment had vanished. The documents, the videos, our half-written paper, gone. All of it. Our access to the installations was revoked and we were forced to sign an NDA by the Ukrainian government, which no one translated for us, and we were deported. The official explanation was expired work visa. It's been almost a year since I saw Luca or Johann. I tried to keep in touch with Johann, but he stopped replying me a couple months ago. I know nothing about Luca and he doesn't answer the phone. Where is their curiosity? Why not chase this further? There's one thing that still bothers me when I think about that asterisk. It was pregnant, wasn't it?